Hello children, how are you? I hope you all are fine at home. Today I am taking the second part of strategies for enhancement in food production of 12th standard and topics, and the topic for discussion are apiculture, pisciculture, plant breeding and its steps, wheat and rice, millets, plant disease resistance, and at last mutation breeding. So let us start. First topic is apiculture, that is also called as beekeeping. So what is apiculture? Whenever we are culturing honeybees on large scale, then it is called as apiculture. And this apiculture is for products, that is for honey and for bee wax. So what are the two products we are getting out from the bees? So they are honey and bee wax. As you all know that honey is very nutritious and it acts as a source of energy and has lot. Wax obtained from bees are also used for making polishes and used in medicinal preparations. Different varieties of bees are, so the local Indian varieties of bees which are used for honey production are Indian bees, rock bee and little bee. And an Italian variety of bee is also being used for large scale production of honey. So these are the different, we are using it for production of honey and bee wax. Now how the honey is obtained? Actually the bee collect nectar from flowers and they convert it into honey. So how the honey is obtained? The honeybees collect nectar from the flowers and they convert it into honey, and that honey is into beehives. Bees are artificially grown in a structure that is called as apiaries. The honey from that apiaries are extracted by a machine, and that machine is called as honey extractor. So artificially where the honey is obtained, or the bees are artificially grown in apiaries and honey obtained is extracted by machine. The name of the machine is honey extractor. The most common species of honey bee is Apis indica. What is it? Is Apis indica. Now let us tips for beekeeping. First is knowledge of habit of bees. Which food, which types of leaves, which types of flower it prefers. It depends upon the types of the bees. So we should know its nature as well as habit of the bees. Selection of suitable location for keeping of beehives. The beehive should be located at certain places where large number of pastures are present. Catching and hiving of swarms. Swarms is the group of bees. First we have to collect it, catch it for starting apiculture. Then management of beehives during different seasons, because there are different seasons. So we have to manage the beehives. Handling and collection of honey and wax. How we will collect honey and wax, so that handling we should know about this. As we know that bees are good pollinator of sunflower, brassica, apple and pear. So thus it increases the efficiency and improve the yield also. So that hive should be kept in such a places where these plants are present. Here you can see the apiaries and beehives where the bee are present. Now the next is about pisciculture. Pisciculture, culture and other aquatic animals on a large scale. So that is called as pisciculture. And this we know that fish is a source of animal protein in our food. Why? Because it contains large of protein. So this pisciculture includes processing or selling of fish, shellfish or other aquatic animals like prawn, crab, lobster, edible oysters, etc. So these are the some aquatic animals which are cultured. So they are called as pisciculture. Different types of fishes are present in different water. First is freshwater fishes, and Catla, rohu, common carp, they belong to freshwater fishes. Next, sardines, mackerel, they belong to marine fishes. In the coastal area, Indian economy as well as employment to millions of fishes totally dependent on this pisciculture. They collect fishes, they sell fishes, they earn money from them and they complete their livelihood from these fishes only. Now let us see what are the difference between pisciculture, mariculture and aquaculture. First is pisciculture. Pisciculture means fish farming in treated water bodies. Mariculture means farming of aquatic organism in coastal water, sea water for commercial uses. And aquaculture means when culturing aquatic plants and animal is done in freshwater body. Then it is called as aquaculture. Here you can see that how the pisciculture is done. Now the next is about plant breeding. Plant breeding is a branch of science, particularly botany, which deals with the alteration of genetic patterns of the plants. And by doing this, we can get hybrid disease resistant variety. So it is the of economic importance for human welfare. So it is a branch of botany that changes the genetic patterns, alters the genetic patterns of the plant. As a result, it increases their values and utility for human welfare. This breeding involves hybridization of pure lines and selection of plants with desirable qualities. Now let us see what are the benefit of plant breeding. First is we can get high yielding variety HYV. So high yielding variety may be obtained by plant breeding. Better quality of the produce. What we are obtaining, they are of better quality. They are resistant to environmental stresses, means they have capacity to tolerate the different environmental stresses. Next one is the outcome of plant breeding. They are resistant to pathogens, means they are disease resistant, and also they are resistant to pest and insect also. Here you can see this is the some Indian hybrid crops of maize, wheat and garden pea. Now let us see what are the main steps of plant breeding. So first is the collection of variability. Collection of variabilities means collection of different wild varieties, species and relatives of the cultivated species, so that we can get all the germ plasm, that is the collection of germ plasm. So whatever the genes available in the population, we have to collect it. So that is called as collection of variability. Second is, after collecting, evaluation and selection of parents. So the germ plasm which we have collected are evaluated to identify plants with some important characters. We will select it, and the selected plants are multiplied. We can get number of seeds in the process of hybridization, and by this we can get the pure line. Third one is, after getting this pure line, cross hybridization among the selected parent. Means hybridization is done between the parents. The selected plants are hybridized. So the hybrid progeny contain both the desirable character of male and female. So both desirable character of male and female are combined together. So the progeny must contain both the character. So that is called as hybrid progeny, and it can be obtained by combining both the traits. This hybridization is a time consuming and tedious process. So sometimes the combination of desirable character may not be obtained. So we have to do it again and again. Next is selection and testing of superior recombinants. So whatever the individuals with desired combination of characters they are present, we have to select from among the progeny of hybrids. Means what the hybrid progeny we are obtained, so we will again select from that progeny of hybrids, and such hybrids are superior to both the parents, means male and female. So that hybrids must be superior to their parents. So they are called as hybrid vigor or heterosis. Now what is next step is, they are self-pollinated several generation till they reach homozygosity. Again we have to find the pure line, inbreed line. So that is why the continuous self-pollination is done. As a result we will get the pure line. So why this is done? So that there is no segregation of other characters in the progeny. Now next is the testing, release and commercialization of new cultivars. After that the selected lines are evaluated for their yield, either they are high yielding or not, and they have some other agronomic characters or traits or not, they are having disease resistant or not. So for this they are tested, then they are commercialized. So evaluation is done by growing these plants first in field and recording their performance under ideal conditions of irrigation, application of fertilizers and other crop management practices. After that they are tested in the farmer's field for at least 3 growing season. How many? 3 growing season at several locations in the country where they can grow. So this will give them different agricultural climatic zones. The material is evaluated comparison to the best available local crops. So they are compared. So suppose one crop is best and the hybrid should be more better than the existing local cultivar. So that local cultivar is taken as a reference material. Then it is certified and the new variety is released. Now let us see about the wheat and rice. These two are which are grown in our country. Production of wheat and rice increases due to the development of semi-dwarf varieties. How it has been increases? Due to the development of semi-dwarf varieties. For this, Norman E. Borlaug developed semi-dwarf varieties of wheat at the International Center for Wheat and Maize Improvement in Mexico. And side by side, semi-dwarf rice variety were derived from IR 8 and Taichung Native 1. High yielding and disease resistant varieties of wheat like Sonalika and Kalyan all over the wheat growing belt of India. And high yielding semi-dwarf varieties of rice like Jaya, Ratna were also developed in India. So these are the some disease resistant, some good varieties of wheat and rice. Now next one is the sugarcane. Saccharum barberry, which were originally grown in North India, but they had poor sugar content and yield. And Saccharum officinarum, which were grown in South India, but had thick stem and higher sugar content. Okay, but did not grow well in North India. So these two species were taken. One was grown in North India, but poor sugar content, and one was grown in South India but has thick stem and higher sugar content, but they did not grow well in North India. So both these species were successfully crossed to get sugarcane varieties with high yielding, thick stem, high sugar and ability to grow in North India sugarcane. So you have seen here that both the characters are combined in one variety. Next is the millets. Hybrid maize, jowar and bajra have been successfully developed in India. Several high yielding varieties resistant to water stress are released. So several varieties of these millets in that it comes maize, jowar and bajra. So they were successfully developed in India. Now next topic is plant breeding for disease resistance. So what are the advantages of disease resistant whenever we are developing disease resistant variety? So first is that we can increase the production yield by reducing, and also second one is reduction in the use of that chemicals like fungicides and bactericides. So these two are the advantages of plant breeding for disease resistance. Resistance of the host plant is the ability to prevent the pathogen from causing diseases and is determined by genetic constitution of the host plant. So how it is determined by the genetic constitution of the host plant. So genes and before doing the breeding purpose. So before breeding, it is important to know about the causative organism and the mode of transmission. Here, this is a table which indicates the name of disease and their causal organism. First is white rust of wheat, that mustard, that is caused by fungus. Brown rust of wheat, that is caused by fungus. Red rot of by fungus. Nut disease caused by fungus. Late blight of caused by fungus. Black rot of crucifer, causal organism is bacteria. Disease, blight of rice is also bacterial. Citrus canker, that is also bacterial. Tobacco mosaic, turnip mosaic and potato mosaic, they are caused by viruses. Now, method of breeding for disease resistance, that is also called as conventional breeding, because always we do the plant breeding for this purpose. So again it includes screening of germ plasm for resistant sources, means collection of germ plasm and their resistance to diseases. Then hybridization of selected parents, again selection and evaluation of hybrids, and at last testing and release of new varieties. These are the common steps for any. Here again there is one table that indicates crop, its variety and resistance to diseases. Variety to leaf and stripe rust hill bunt. Brassica, that is Pusa swarnim or currandrai, that is resistant to disease white rust. Cauliflower, that is Pusa subra, Pusa snowball K1, that is resistant to black rot and curl blight, black rot. Next one is cowpea, variety is Pusa komal, that is resistant to bacterial blight. Second one is next one is chili sadabahar, and it is resistant to chili mosaic virus, tobacco mosaic virus and leaf curl. Now last is the mutation breeding. It includes mutation through various mutagens. Mutagens means those that chemicals which are responsible for creating mutation. Mutation means it can be abrupt change in genetic constitution. So that is mutation. Can be done by inducing or by with the help of mutagens, and mutagens may be physical or chemical. So what here also is screening the plant material for disease resistant. First we have to select plant material, then we have to multiplicate, that is multiplication of these selected plants for direct use or for use in breeding. Now next is hybridization of selected plant, then selection for disease resistant, again at the last testing and release of a variety. These are the common every breeding. For example, in mung bean, resistant to yellow mosaic virus and powdery mildew were induced by mutation. So thank you children. This is all about. For any kind of doubts, please section.